Well, okay, I'm giving you a warning. That's why, cause I wasn't aware. And it was bad. It was so cold.
I can only imagine. Okay, so just for clarification, no matter where you are on Friday, just because we have a lot going on, that lecture 10 quiz is due on Friday. Also, technical paper is due in on November 23rd, the first Sunday of Thanksgiving break. Again, that is the last possible day to turn it in. Okay, last possible day. And then for exam 3 is next Tuesday. On Friday, we are moving forward with the crash course meiosis just because we do have to, like I said before, we only have 3 weeks left. Again, not including this week, we only have 3 weeks of material, and we still have to get through exam 3 and exam 4. Exam 4 only is 11, 12, and 13. 11 is pretty fast, 13 is pretty fast, 12 takes a little bit of time, but it should be fun, okay? So, any questions on that before we move on? Okay, so again, we are gonna use our Chromebooks, but the last 10 minutes of class, so we go ahead and just keep them at a 45-degree angle, but let's go ahead and move on. So we did talk about the first part of transcription on Friday, and remember we watched the video on page 10.5, 10.6, and I also said that was homework, right? Whenever you didn't finish. Yeah, okay. That means I said it regardless. But so just as a reminder, when we talk about RNA, remember there are 4 kinds of RNA. What is the first kind? mRNA, messenger RNA. And then in addition to that, just again for clarification, we only have today, or not including today, Thursday, Friday, next Thursday, next Friday, next Monday, Wednesday, Thursday, Wednesday, Friday. We only have 9 more lectures, like days that we're actually teaching, okay? Until the end of the year, only 9 days. So just for clarification, I want you to make sure that you put in 110%, 9 days, that's it, okay? Because after spring break, or Thanksgiving break, we're reviewing and final and everything. I'm removing, and that's it. And then after we finish TJC, I'm done. Be quiet, I think I said it before, right? Y'all can make deals. So, messenger RNA, but what is messenger RNA or mRNA? What is it? One word. It's just a copy, that's it. tRNA, it's a little bit different, but it has a little piece of what attached to it? It's an amino acid that's attached to it. And ribosomal RNA is a little bit different too, because when I connect or combine our RNA with protein, I end up getting a ribosome. snRNA is really new to us because you didn't talk about it in high school biology, but snRNA is what exactly? Well, snRNA, when I mix it with a little bit of proteins, kind of like a ribosome, I make a spliceosome. Spliceosomes do something a little bit different. Again, today we'll talk more about it, but splice means to join or to glue together, okay? So, also, what is a stop codon? A codon is 3 letters with 3 bases that tells the ribosome to stop. Start codons are 3 letters with 3 bases that tell the ribosome to start. But stop and start what? Translation. Stop and start what? Translation. But translation is to make a protein. Just like transcription is to make.
Excuse me, teachers, staff, and students for this interruption. All juniors and seniors attending the JA trip to Orr this Friday, please come to the chocolate closet to grab your kit. Thank you.
You really win?
Where are you going? Oh yeah, you told me.
Alright, anyways, so we got that, and we already did this, and so now we're gonna look at transcription. So if you would, go ahead and read it for me, please. Sandy, go ahead and read the first step of transcription, or initiation, sorry.
In this process, DNA is copying into RNA.
Well, initiation,
RNA polymerase binds to a promoter region called a TATA box on the DNA connected strand.
And then, so it is referred to as a Tata box, that's just kind of what they're called. But these are what we call promoter regions, where we basically start, okay? What does it do, RNA polymerase, what does it do after it attaches to the promoter region? Go ahead and read that next one for me, please. Abraham, the second bullet point.
RNA polymerase breaks the hydrogen bonds between bases and two strands separate.
So the first step is that RNA polymerase binds to a what? And the second step of initiation is that it just breaks the what? Hydrogen bonds, good. And then elongation. What does elongation mean? If initiation means to start, what does elongation mean? To continue or to make longer. But what are we making longer? The strand of? Not DNA, but RNA. Making the strand of RNA longer. Go ahead and read the first bullet point underneath elongation for me, please. Caitlin.
RNA polymerase reads the template strand and builds an mRNA molecule with complementary bases in the 5 to 3 direction.
What does that mean, complementary bases? Can anyone tell me what they think that means?
The bases that match up with each other.
The bases that match up with each other. Give me an example of
Adenine,
Say what method?
adenine and thymine.
So adenine and thymine. But if this were thymine, what would it put it in its place? If red on DNA, thymine, it would match up with what? Does it? Thymine right here on DNA, what would it match up with? Adenine. And if adenine was right here, what would it match up on RNA? Uracil. One more time. On DNA, if it's an A, what is it going to be on RNA? DNA is A, RNA is? What did I say? If DNA is A, RNA is? Uracil. Good. If DNA is guanine, what is it on RNA? Guanine, cytosine. If DNA is cytosine, what is it on RNA? Guanine. There we go. Okay. So first step is called what? The first step of everything is called what? Which means to start. Elongation means to make longer. So in initiation, the RNA polymerase hooks onto DNA, but what region? One more time. In initiation, RNA polymerase hooks onto DNA, but what region? The promoter region. And then it cuts what? Hydrogen bonds. Then after it cuts the hydrogen bonds, then it reads DNA and just matches up the complementary bases. So if it's an adenine, then a uracil, thymine, adenine, guanine, cytosine, cytosine, like on and on and on, okay? But in the what direction though? 5 to 3, just like DNA was made in a 5' to 3' direction, okay? Then the last bullet point underneath elongation, again, the last bullet point underneath elongation, go ahead and read it for me, please. Sorry.
For example, if
The last bullet, this continues.
this continues for the length of gene.
So, again, elongation lasts as long as the gene is what? Elongation lasts as long as the gene is going. So for example, if the gene is this long, then elongation is this long. If the gene is this long, then elongation is this long. If the gene is this long, elongation is this long. Does that make sense? Because it just depends on how long the gene is. Because it depends on the protein, okay? Then the last step called termination, what does that mean? Go ahead, or what does termination mean in general? It means to stop or to end. But what are we stopping or ending? The process of, this whole thing is called what? Transcription. Go ahead and read that bullet point for me, please, Maggie.
Transcription ends when the RNA polymerase reaches a terminator sequence in the DNA template strand.
So, in other words, right here, looking at this image, RNA polymerase hooks onto what region? Promoter region, and then this region, which is called the transcribed region, the RNA polymerase just reads the DNA and matches up the complementary what? Bases. again, RNA polymerase goes through this region called a transcribed region, because it's the region that we're actually, what is transcribed? Transcription? To write or to copy. So one more time. This region is called the transcribed region, and RNA, as it moves through it, RNA polymerase just matches up the complementary bases. And it's gonna keep doing it as long as the gene is long. But when does it stop? When it reaches a terminator sequencer region. Okay, so let me go ahead and write that. So letter D, this region is called the what region? Promoter region. Okay. H, say what?
What did I miss?
A lot. H is called the transcribed region. And what is I?
Termination.
Termination sequence or region. Did you figure your journal method? I'll print off the last paper that we're gonna do today so you can get it done and just staple it together. What is this entire thing? These parts right here that branch off. What is this? Strand of what? Not RNA, but a strand of DNA. What is F? Strand of RNA. What kind of RNA? Because there's 4 kinds that we discussed. mRNA. So this strand of DNA, this part, this region, this sequence, what is it called? That's the step, but what is this region or sequence? The promoter region. This entire region is called the what? Transcribed region, because that's the part that we are copying, transcribing, transcripting, okay? And then this last section is what we call the what? Termination sequence. So again, in the first step called initiation, what happens? RNA polymerase hooks onto the what region? Promoter region, and then it cuts the what? Hydrogen bonds. The second step called what? Elongation, we're making what longer? What strand? Of RNA. We're making the strand of RNA what? Longer. But how do we do that? RNA polymerase reads the DNA and copies down the complementary bases. So if this is a thymine, what should it be on DNA? Adenine. If this is an adenine, what should it be on RNA? If this is cytosine, what should it be on RNA? 1. Just like that. Okay. Every 3 letters on DNA is called a triplet. Should sound familiar because every 3 letters on RNA is called what? A codon. Again, every 3 letters on RNA is called a codon, but every 3 letters on DNA are called a what? Triplet. Triplet because it's 3 triplets. Okay. Does that make sense? Okay.
Oh no!
Okay, so I'm gonna wait. You're not going?
He said the bus was already full because we're going to the A&M trip, so they only have 2 buses. So we have to wait till next time, unless we really, really want to go.
For what?
Unless you really, really want to go, pues don't tell the channel to
they're gonna like take a few people off the bus and make them drive.
Oh,
So,
the J trip?
Mhm.
Huh.
Well, I thought we couldn't drive because of liability now. That's what Mr. Rose told me.
I don't know, not my problem. Anyways,
Anyways.
so again, so since a lot of us missed it, we're gonna do it one more time, and then we're gonna move on, because we ain't got time, okay? Because again, most, raise your hand if you're going on a trip on Wednesday, or Thursday. One, you're not going, Nicholas? 1, 2, 3, 4, 5. That's it?
You expect there to be a lot more, right?
Yeah.
a lot of,
It's a scam.
Well, no, because she changed it. If you're, as long as you're passing high school classes, you can go on the A&M trip. So it's probably gonna be more now.
Nicholas, do you wanna go?
Oh.
Sarai, do you wanna go? Honestly, Sarai, you're like the vibe. I wish I could just like live like you. But okay, anyway, so as a review and for the first time, transcription has 3 phases: initiation, elongation, and termination. Initiation means to start, elongation means to make, and termination means to stop.
Thank you.
But when we look at the process of transcription, it starts off with C, which is RNA polymerase attaching to a specific area called the promoter region. One more time. RNA polymerase attaches to the promoter region. And then after it attaches to the promoter region, it cuts the hydrogen bonds. Then RNA polymerase moves through the H, which is the transcribed region, which is the part where it's actually making a copy of it. So as it moves to the transcribed region, it matches up the complementary bases. So whatever's a triplet, the codon should be. Remember, a triplet is the 3 letters or bases on DNA. Codons are the 3 letters or bases on RNA. So as we said, if DNA has thymine, RNA should have adenine. If DNA has adenine, RNA should have uracil. If DNA has cytosine, RNA should have guanine. Okay? And there's a specific reason why I put AUG first. Okay? Because it's a start codon. Okay? Does that make sense? Okay. Then the RNA polymerase is just going to keep making an RNA molecule until when it reaches a termination sequence. When it reaches the termination sequence, then it stops. Doesn't actually produce anything, it just tells it some letters that tell it to stop. That's it. Okay? But at the end of this, we end up getting F, which is a piece of yes, mRNA, but what kind? Immature. Because we have to do a couple things to it. Okay? Speaking of which, where does this entire process occur? What organelle? The nucleus. Why the nucleus? That's where DNA is located. If this were a prokaryote, where would it occur at? In the nucleoid.
Oh.
Good. Very good. Very good. Any questions, or do you want to go through it one more time? So again, remember that RNA polymerase attaches to the what region? Promoter region. Once it attaches, it cuts the hydrogen bonds. After it cuts the hydrogen bonds, then it moves to the what region?
Transcribed region.
Where it just matches up the, social the scene, complementary bases. And it does that and what is this step called? Where it moves to the transcribed region? Elongation, because what is it making longer? The strand of mRNA. And then it's going to continue until the RNA polymerase reaches the termination sequence. Once it reads it, then it stops. It breaks off, and the piece of product that we get is called a what?
Immature mRNA.
Immature mRNA. Okay, so we gotta do a couple things to it. Okay. So what do we actually do to it? Well, we do 3 things. 1, we put on a guanine cap. So what's one thing we do? Add a guanine cap, and if you look and did page 10.5, 10.6, on page 10.6, it actually shows like a little hat in there. Okay. Then what else does it do? Not only does it put a cap on it, but it puts on a
Polyated.
polyatel. Okay. Why does it put on a cap and a tail? Because again, where does this process occur? Transcription?
And then the nucleus.
And the nucleus. Why is DNA in the nucleus in the first place? 2 starts with a P. To protect it. So as soon as we send that DNA out, it's unprotected. So we protect it by putting on a cap and a tail. One more time. DNA is protected in the nucleus, but since we just made a copy of DNA in the form of mRNA, we want to protect it, so we put the 5' guanine cap, poly A tail, and then we send it out. Okay? But before we actually send it out, we gotta do something else to it. Okay? Now, if you look, there's these regions called introns and these regions called what?
Exons.
Introns, go ahead and read it for me, please. Savannah, introns do what? Jonathan, exons do what? Express. So introns are interrupt exons, and exons do what? Are express. Again, exons are express. That's why they're called exons. Okay. What do we notice from this top picture to this picture? What do we do to the introns? What do we do to the introns? Do we leave them?
No, we take them out.
We take them out and we glue the pieces back together. What pieces?
The exons.
The exons. But what does the word excision mean? Again, in the thing it said it, but what does excision mean?
To cut out.
To cut out. What does splice mean? To join together. So who does this cutting and splicing, or excision and joining together? The spliceosome. That's the whole point of it. Okay. Remember, a spliceosome is a little bit of snRNA and a little bit of
Protein.
proteins. Again, it excises or what?
Excises are cut out.
Excise or cuts, but what does it cut out?
Cuts out the intro.
Because they interrupt. And it glues or joins or splices the what?
Exons.
Because those are the ones that are
Express.
expressed. And so this mature mRNA has a 5' guanine cap, has all the exons, and the poly A tail. Now we can actually send it out and actually do its job, which is make a protein. Does that make sense? So what are the 3 things that happen to immature mRNA before we send it out to actually make a protein? What 3 things? We had a 5' guanine cap. We had a poly A tail, and 2 or 3, we excise or cut out the because they interrupt, and we splice or join together the exons because those are the ones that are expressed. But who does all that excision and splicing? Spliceosome. And that's it. It's ready to be made into a protein. Okay? Does that make sense? Okay. So you are gonna use your Chromebooks. I'm gonna go ahead and give you guys 26 minutes-ish. We'll see where we are with that. To go ahead and do this page, which is page 2.7 and 10.8. And then Matthew, I'll go ahead and print this up. Okay. You feeling okay, Matthew? Don't feel good though? Matthew, I printed it to the printer in the nest. Just go ahead and go pick it up.
figured it out, and I was like, sure, I guess, I don't know. Like,
I don't see it.
the response?
Yeah.
What? Let me show you.
He probably,
He probably only responded to me, but he said your name.
okay.
I'll show you. I've like never, he's like never received something before, so he just told me, that's why I.
Jonathan, Nicholas, focus. Christian, you too. Oh, last year? No, I remember that. Actually, I don't know. What was it about? And she just like that. Oh, yeah, I remember.
It's terrible.
That's good.
I know. I was like, look at you, Miss Professional.
Oh, you did 16, so I don't know why you did it.
It's probably drowning the rest of everyone else's. Was there anything else that you said about when we were doing that? I can't remember.
Did you send it to my TJC email? Oh, that's why. I didn't see it.
Oh.
That's why. I probably said.
Yeah, that's the first one that came up whenever I typed in like your last name, your first name. That's the first one that popped up. Cause I did it through my TJC email instead of Tyler Institute.
Got it. Which one are you on?
Was there anything else? Cause this is basically everything you put on.
That's basically everything.
Yeah.
And that, and just looking at the structure of a synovial joint.
Okay.
But the biggest thing was knowing that it's like, kind of like a present, because it's all these different layers. So it's the ligament, then the fibrous layer, which is literally just, it's regular connective tissue. Then it's the synovial membrane, which only covers the sides. Then the articular cartilage on the edges, and then the fluid in the cavity space. And that's basically it.
I'm gonna come on Monday, cause I need to refresh all on 7. It's like, it's here, but it's very far.